It looks like it's recording. Okay. So the first thing I'd like to do is point everybody to the Google spreadsheet that we've been collaborating on. One of the, I think, most exciting part is that we're starting this resource worksheet where we're all including what we work on or what is available out there. And feel free to use it and add to it. And there are names next to the different types of models or programming languages. And these names are basically telling you who you can contact if you need any help or advice in the field. So for instance, I use Mathematica for most of my work. And so you're always welcome to ask me for this. The other thing, we also have a Slack channel. So if you would like to join or you haven't, just contact me and then we can go. Does anybody have questions? No? Does anybody want to introduce themselves before one more person? No? Okay. Oh, I hear. No. Okay, well, I guess the best thing is to get started. And I'm really happy that Marc has agreed today to give a talk. I'm actually looking forward to hear what it's about because I do not know very much about graphical models, but it definitely sounds exciting. Thank you,
You're gonna
Marc.
have to let me share. Right now I can't share.
Oh, sorry. Yeah, let's see.
Yeah, it's okay.
Sorry. Is it working?
Yes.
Okay, great.
Now I just have to find the right window, and there we go. And okay, there we go. Well, there we go. So I'm going to try to give this talk without going into too much technical detail. What I want to do is kind of give you an idea of some techniques that are out there that you can use to try to figure out what is the bit of your model that's responsible for the interesting behavior. And I'll explain in a minute what I mean by interesting behavior. And it assumes, of course, your model has some dynamics that's of interest. So the problems with cell biology kind of as it exists today is that we often have really large models. We have often really good information, or at least really strong hints in the literature as to how things are connected together, right? I mean, this gene, when it gets turned on, makes this protein. That protein, goes and does something, which eventually through some signaling cascade propagates back and turns on some other genes, right? So those kind of details of how things are connected increasingly are reasonably well known. That results in large models, which, of course, there's ways to boil things down, but nevertheless, we often have a lot of connections in these models. But on the other hand, parameters are a problem. And I think this has been mentioned once or twice in talks before. There's often not that many measured parameters, rate constants, protein concentrations, never mind things like, rates of transcription or translation or anything like that. Often a lot of that stuff, again, there's scattered pieces in the literature, but when you try to work out, a specific model in detail, often you find a whole bunch of parameters are just not known. And even worse, even when you can find parameters, they're often not transferable. The parameters, rate constants, for example, are often quite different from one organism to another. Certainly protein levels are very different, so you can't take data from a mouse and they won't necessarily, they may give you kind of an order of magnitude, if you're lucky, kind of estimate of a parameter, but often not even that. And so parameters from mouse may not tell you much about exactly what the parameters are in humans. Never mind going farther away from, right? I mean, anything that happens in E. coli, even when we have similar enzymes in mammals, tells you those are so different. There's no information there that you can use. Um, there are some good databases out there that contain some stuff. So enzyme kinetics in particular, it's not too bad that way. But even then, if we can ever get somebody from Santiago Schnell's group to come join us sometime, they'll tell you that, the the kinetic parameters are so dependent on the conditions within the cell that often, measurements in vitro tell you almost nothing about what happens inside the cell. So, um, so we have this problem that we have large models where we kind of know the shape of the thing, but we often don't know the parameters. So you'd like to be able to say something about your model without necessarily knowing what the parameters are. Um, so um, this is a discussion series. Oh, and I should say it is supposed to be a discussion series. So interrupt me with questions anytime, right? So this is a discussion series on cell and developmental biology, right? If you think about the things that cause developmental events, some of them are associated with clocks, right? So there's the well-known semitogenesis oscillator, for example. So we have that kind of dynamics that's interesting to us. Um, we Multi-stability, of course, is what makes multicellular organisms possible, right? You have one genome, but there are many different stable cell fates that you can get from one genome. And of course, at smaller levels, that's an extreme example. At smaller levels, an awful lot of the local decisions about what a cell is going to do are based on having two or more possible steady states and the cell choosing one. But more than that, the bistability during development, right? So the bistability often results in waves, for example, waves of morphogens. And so we have those kinds of dynamics as well. And then, of course, everybody talks about Turing patterns. There's relatively few, even after all these years, relatively few examples where you can definitely say, that's a Turing pattern. But people still invoke Turing instabilities as explanations for developmental patterning. So we have all kinds of interesting dynamics that might be important in development. That's certainly important in some developmental events. And again, we'd like to be able to maybe connect dynamics to maybe the structure of the network without necessarily, again, having to know all of the parameters. So the general question I'm going to um, give you a kind of a broad brush picture of today is, are there network features that give rise to instabilities, and what are those network features, and how can I find them in a particular model? So this actually takes us to some really old research, and this is a highly incomplete list and highly idiosyncratic as well. And there are holes in here that I'll fill in on the next slide. But there's a long history of qualitative stability analysis going back at least to the 1960s. The Quirk and Ruppert paper, by the way, is in economics. Um, but lots of people over the years have made interesting contributions to this. There's some nice papers by René Thomas, the late René Thomas, and Marceline Kaufmann in Belgium. Um, uh, Martin Feinberg has done a whole lot of work, some of which I'll touch on again in a few minutes. George Krasien is kind of an emerging star in the field. Pauline Vendendriesch, whom many of you know, I'm sure, who's at the University of Victoria, has done some interesting work in this area too. So there's some work, some old, some new, on trying to use qualitative features of a network to say something about the possible dynamics of that network. Now, one of the reasons there's a hole in that slide before is because a lot of these methods are based on graph representations of, in particular, chemical networks. And of course, the cell is kind of a big, at least from my perspective, the cell's a big chemical network. Um, and this stuff goes back at least to the 70s with Bruce Clark, who was at the University of Alberta for many years. Contributions by lots and lots of people. You see Feinberg's name again in there, and George Krasien again, and several others. I'm going to follow a particular line through this literature. Well, in fact, I won't tell you the whole history, but the line that I'm following starts with Bruce Clark in the 70s, and then it was followed up by Ivanova, who wrote these little papers in Russian that are really intriguing but devoid of any proofs, and kind of not not entirely satisfying, I guess, in some ways. And then we picked up this work. Well, Maya Mincheva, who came to my group as a postdoc, really kind of brought this idea that she wanted to work on this, and we published a series of papers where we basically we took Ivanova's work and we put it on a solid footing with kind of detailed proofs of why this works and so on. Maya's followed that up. She's still working on a lot of this stuff. So that's kind of the so that's just a quick historical kind of thing again. So just to situate us, we're interested in qualitative stability analysis methods, and specifically ones that have a graph representation. And I'll explain in a few minutes why we want a graph representation. So um, a 1 minute overview of where I'm going with this. So I'm going to call it the Ivanova family of methods because it started with Clark, but Clark's methods are they're a little different. They've not Ivanova adapted some things, made them a lot easier to use. So these methods apply specifically to mass action systems. So in other words, systems where you write things in terms of a set of elementary reactions and then use the law of mass action to write down the differential equations. Um, if you don't have a mass action system, it's generally possible, unless you have really weird nonlinearities, it's generally possible to cook up a mass action system that would have your system as a limit and a singular perturbation limit. So it's not as bad a limitation as it sounds, although going from a kind of a non-mass action to a mass action system does make the system bigger. So roughly speaking, what we're going to do is we're going to build a bipartite graph, and I'll explain in a couple minutes what that is. But a bipartite graph that represents the reactions in the system, and we're going to look for specific structures that turn out to be associated with instabilities. Um, what we'll get out of that is a fairly surprisingly simple necessary condition for saddle node bifurcations. And for those of you who are maybe not so much in the bifurcation theory area and nonlinear dynamics, a saddle node bifurcation is what you need to get multi-stability. So if you're going to have two or more steady states, except for some oddball systems, generally speaking, you'll have a saddle node bifurcation that creates the extra steady states. Um, we also get a very simple, just a simple a condition for Turing bifurcations. So pretty easy to tell using these methods if a Turing bifurcation is a possibility. Um, and for Andronov-Hopf bifurcations, unfortunately, there's two cases. One is simple and one is not so simple. And I'll talk about the simple condition, and the not so simple one, I think I'll refer you to Maya's papers. Um, so um, in a nutshell, that's kind of the main thing. And then at the very end, I'll just mention really briefly that there's an interesting extension of these methods that treats delay-induced instabilities. And that's really interesting for us because, of course, gene expression, um, there's necessary delays between when you induce a gene and when you finally get a product, right? Transcription, if you think about eukaryotes, for example, the genes in humans, okay, so it depends on which eukaryote you're talking about. For the yeast people, you live in a blessed world where things are a little simpler. But for humans, we have massive genes that have many introns that have to be spliced out, and it's a very slow process. It can take anywhere from tens of minutes to hours, literally, to just get an mRNA, right? So there are significant delays in these systems, and being able to say something about whether those delays can induce instabilities that would not be there in the absence of delays is an interesting thing. Okay, so
I've got
by
a
part,
little question.
yes.
So I'm by no means an expert, but is this somehow related to chemical reaction network theory? Because this is something that I've heard about, so I'm just trying to make a mental connection.
Yes, so it's a real good question. So chemical reaction network theory is the stuff that Martin Feinberg started to develop, I think in the 80s, but it has an even older history than that. It goes back to Horn and Jackson in the 70s. Um, uh, our methods are different, but they have some contact points, and there are the kinds of things you can say about the systems are have some they're very similar. So all of these things give you necessary but not sufficient conditions for various types of instabilities. uh, they use different graphs, and the some of the results that you get from one theory or the other are stronger in some cases, but they seem to be, as far as I can tell so far, and we haven't done a really detailed comparison, but it seems like the methods are somewhat complementary. So cases where, kind of we run into a brick wall because they're in one way or another kind of too hard. Often chemical reaction network theory will give you some information. There are other cases that they have trouble with that we don't.
Hmm.
So they're kind of complementary to some extent. And later on, I'll mention too, there's software to do some of these things. Okay,
Okay, cool.
great. All right. So graphs. This is the proverbial toy model. This is the Brusselator, which you may have heard of. It's an old model that came from Prigoghine's group in Brussels, hence the name, the Brussels oscillator. It's a portmanteau, right? At one point in chemistry, there was a real kind of wave of people naming their oscillators and their oscillating models for where they were working. So you got the Oregonator and the Urbanilator and all kinds of other awful things. As I always tell my students, I don't think I'd ever name a model the Lethbridgeator. It just sounds terrible. But anyway, so it's a little oscillating model, real simple, four chemical reactions. Two of them are just kind of X reactant flowing in and then being degraded or flowing through the reactor or something like that. The real interesting reaction here is this one, which is an autocatalytic conversion of a Y, um, autocatalytic conversion of a Y by X to another X. And so this model, if you've ever, if you haven't studied it, I guess I should tell you. So it's an oscillator. It has, you can get Turing patterns out of it. So it has a lot of the kinds of things people are interested in. I see there's something in the chat, but the little chat thing keeps disappearing on me. I guess, oh, hello, Widodo. um,
So somebody said
oh,
in chemical reaction network theory, the graph is
that's
not a
right.
bipartite graph.
That's right. That's right. In the classic chemical reaction network theory, they have these things called complexes. So for example, 2x plus y would be a complex, and the thing connects up the complexes. Um, there's kind of a, there's a system for
They're
weighting
called
edges and stuff. Hmm.
species. So you have the species set, complex set, and the reaction set. So the XY are the species set, and 2x plus y is one complex, 3x is another complex, and then you have the reaction. So the reaction will be X going to Y, that's one reaction. So you have three sets. So it's a
Yeah.
3 to 4 system.
Yes. Yes, that's exactly right. So yeah, so they use different graphs, and they use and that's right, they use multiple graphs in the end. I should say too, the theory's been still under active, the chemical reaction network theory is still under really active development, and there's versions now that use a bipartite graph, but not the same one that we use. So there's lots of graphs floating around in the literature. So anyway, how you translate this thing into a bipartite graph is that your reactions, which I'm representing by V's here, sometimes we use R, your reactions are one type of node, and then your chemical species are another type. And essentially you have an arrow from a chemical species to a reaction if it's a reactant, and from the reaction to the product, that's it, if it's a product, right? So there's a real simple relationship. You can read these and turn them back into chemical reactions pretty easily. We weight the edges when they're stoichiometric coefficients. Implicitly, all the weights are one unless you write a weight. So this reaction here, the third reaction, we'd have 2x plus y. So we've got those arrows coming into this and making 3x. So it's a pretty simple representation of a chemical network. Um,
Can I ask a
so
question?
yes,
Just
of course.
sorry. Just to understand, in terms of the cell, are just gene interactions, or how is this being
So
used?
when we do this kind of modeling, we're often looking at systems where there's a signaling network, there's genes, we have to look at transcription factors binding to genes. So every distinct chemical species, so whether it's a signaling molecule or a modified version of a signaling molecule that's been, say, phosphorylated or something like that, or it's a transcription factor. Well, one of those signaling molecules might be a transcription factor that, or we'll have species that represent the gene promoter when it's empty, and then one, where we've taken a transcription factor, say an activator, and the promoter, put them together. So every chemical species is a node. And then the reactions are literally their chemical reactions, binding events, whatever they are. Okay.
Thank you.
So these graphs can get actually quite large, right? Again, this is very much a toy model. I'll show you something with a more realistic system just a little later. Okay, so why do we want these graphs? What's the point of them? Well, it turns out that there are subsets of the bipartite graph, which we call fragments, which have a one-to-one correspondence with terms in the characteristic polynomial. So if you're going to do stability analysis, you're going to be looking at a characteristic polynomial. And you can extract these pieces, the fragments, that have an exact correspondence to a term in the characteristic polynomial. So what's a fragment? So in a fragment, what you've got is k distinct chemical species and k, I misspelled that, k reactions, but the reactions are not necessarily distinct. So sometimes you go through the same reaction node twice, but you have to have k distinct chemical species. And so if you've got a fragment with k chemical species, it will correspond very directly to a term in the characteristic polynomial. So this way, so your characteristic polynomial, let's say your dummy variable is lambda, your eigenvalues, right? And it's got some coefficients. So the coefficient of the n minus k term, it'll have a bunch of terms in them, and any given one of those terms, you could relate it back to a fragment with k chemical species, we say order k. So you've got k chemical species, and you've got k reactions. Again, the chemical species are distinct. These are not necessarily distinct. And for every fragment you find, you will have a term in the characteristic polynomial that basically what you do is you take the rates of those chemical reactions, again, mass action rates, so they're all monomials, right? So you take these rates and you divide by the corresponding concentrations to the chemical species. And then there's this coefficient. So these KSKs. And that is a quantity that's literally a number. So that's like, it could be 1 or it could be -2, or it's a number. And the whole game is figuring out what these things are, because it turns out that what these numbers are has a real simple relationship to at least the potential for instabilities. So I'll get to that in a minute, but this is kind of as technical as I'm going to make this talk. But so the big picture is there's a formula, there's a prescription for figuring out what fragments you can make out of a system. And it comes down to, and I think I mentioned that in the next slide, it comes down to fragments are composed of cycles and edges. And for every fragment, there's a term in the, or there may be, I mean, that K coefficient can turn out to be 0, in which case the term just disappears. But at least in principle, for every fragment, there's a term in the characteristic polynomial that's part of the overall coefficient of lambda to the n minus k. Oh, and n, I should say, is the number of independent chemical species in your model. Okay, so
Sorry to
yeah,
interrupt again. So this characteristic polynomial comes from the differential equation system.
Exactly right. So if you wanted to develop the characteristic polynomial in detail, you would start with your differential equations. You linearize them around a steady state, and that gives you a Jacobian matrix, and the eigenvalues of that Jacobian matrix. If all their real parts are negative, then you have a stable steady state. So the interesting stuff happens when one of those real parts, or a pair of them, if it's an Andronov-Hoff bifurcation, crosses zero and becomes positive. All right.
And the graph, you can use it to construct the connectivity matrix that you use in the system of differential equations, or am I in a completely different field now?
So the bipartite graph had the bipartite graph is directly related to the rate equations, because you can effectively again, it's mass action, so multiply the reactant concentrations together and times a rate constant, that gives you the rates. And it's this relatively simple relationship that creates this maybe slightly less simple relationship between these fragments of the bipartite graph and then the characteristic polynomial. And the fact that it comes out like this, where you multiply the rates and divide by the concentrations, that's totally a mass action
Mm-hmm.
thing. If you change the type of kinetics, there are other methods, some of which we worked on a little bit, that will work directly on a non-mass action model, but the results are not quite as sharp, for a bunch of reasons that you can imagine. If you have a kind of an arbitrary nonlinearity, it becomes hard to pin down too much about what might happen. Okay.
Mm-hmm.
Okay, so what are these fragments? So they're unions of subgraphs, and the subgraphs themselves are unions of cycles, and then edges, but specifically edges that link a reactant to a reaction. So the edges that go from a reaction to a product, those those can't be part of a fragment. Um, and you can imagine the reason for that, right? Is because the products don't affect the rate of reaction. It's only in mass action, it's only the reactants that matter. Um, and within a subgraph, each chemical species can only be at the origin of one edge within the subgraph. So um, these critical the fragments that matter are the one, or I shouldn't say the fragments that matter, but the fragments I'm going to most talk about are called critical fragments. And they're ones where that numerical coefficient that I mentioned previously is negative. Um, and just to give you a picture of the sorts of things we end up looking for, a critical fragment has to have at least one subgraph with an odd number of positive cycles. So we look for positive cycles, and a positive cycle is essentially one where um, actually maybe it's better if I give an example in a couple of minutes, but uh, in the simplest case, a positive cycle, you're going from reactant, reaction, product, which is then the reactant of the next reaction, product, and all the way around, and you're just following reactant to product relationships. But there's also things called negative paths that show up in these things, and as long as you have a negative path encodes a co-reactant relationship. So you can have those in your cycles, and as long as you have um, as long as you have an even number of these negative paths, you still have a positive cycle. Um, and again, I'm trying to stay away from too much technical detail, because I think I'm trying to get kind of the big picture of what we can do here across. But having said that, sorry, I can't remember your name. The person who keeps asking the great questions.
Erica.
Sure. Meaning me, that's Erica.
Erica, yeah. I mean, yeah. So, I mean, if there are technical questions, I can get into them for Erica or anybody else who wants to. So, okay. So there's some easily applied theorems, not horrible to prove. So the first one is there's a really clean theorem for saddle node bifurcation. So again, remember, saddle node bifurcations give you bistability, and the theorem is that a necessary condition for a saddle node bifurcation is a critical fragment of order n. and N is the number of independent chemical species in your system. Um, so you basically look at the so if you want to think of it in terms of the characteristic polynomial, maybe I'll go back here for a second, right? So this term over here, if you want a saddle node bifurcation, this term has to pass through 0. And that's it. And the only way that's going to happen, so remember the critical fragments are the ones that have a negative k. The only way that's going to happen is if there's at least one term in here that has a negative sign. If all the terms in there are positive, and this is the nice thing about mass action systems, right? So for those of you doing electrophysiology or something, it's not so nice. But for us, you know, the chemical species all have positive concentrations. The rate constants are all positive numbers. And so you can literally see the negative terms in your characteristic polynomial because they're the ones with negative signs in front of them. Maybe I should say one more thing. If you've got a really big system, if you've ever tried to develop the characteristic polynomial for a really big system using Maple or Mathematica or something, it is very crunch intensive, and it's not unlikely that your computer will run out of memory before you're able to even get this polynomial out. If you could do that, you might not need this graph theory. The nice thing about this graph theory is that it turns out to be very nicely parallelizable. And so we're able to do some of these things without ever working out the entire characteristic polynomial. We just find these fragments that have these special properties. So again, so the saddle node case is really clean. You need a critical fragment, and that's that. And you need a critical fragment of order n specifically. Um, if you're going to have an Andronov-Hopf bifurcation, there are actually two cases. One of them is the case that we would normally think of as positive feedback, and that one you need a critical fragment, but of order less than n. I should have said that in here. So any of the previous terms should have a critical fragment in it, and then you might have an Andronov-Hopf bifurcation. But again, the last term, the one that doesn't have a lambda with it, that's that one that gives you saddle nodes. Oh, and I should say, for those of you who are experts in bifurcation theory, saddle nodes plus the degenerate cases, right? So transcritical bifurcations and pitchforks. So now we're just looking for critical fragments, and then here's another nice one. If you're going to have a Turing bifurcation, you have to have a critical fragment. So if you can find critical fragments, if they're order n, you might have a saddle node bifurcation. If they're order less than n, you might have an Andronov-Hopf. Note these are necessary, not sufficient conditions, but then all of these things give you necessary, not sufficient conditions. And whether it's order n or some other order, if you've got a critical fragment, you might have a Turing bifurcation. So pretty cool. Now, here's the bit I want to point out, and I'll show you an example in a couple of minutes, is this already is giving us what I said, right? We want to be able to kind of pick out what's the part of this network that gives us the interesting behavior. Well, the answer is going to be the things that are in your critical fragment, and the interactions encoded in that critical fragment are the things that are critical to the behavior. So critical to getting bistability, critical to getting oscillations due to positive feedback or Turing bifurcation. The case of oscillations due to negative feedback is unfortunately much harder. And if you ask me right now to tell you how it works, I would fail miserably, because it involves finding two positive fragments whose order is separated by 3, and there's a complicated condition they satisfy, where you have to look at kind of a group of things they belong to, and which groups they belong to. They called these things called flux fragments. But to boil it down to kind of the kind of the one thing you might look for before you even attempt this, you have to have these two positive fragments, so non-critical, and according to what we had before, at least one of them has to have a cycle of order 3 or more, or um, or there's no hope. So cycles are really kind of central to the whole thing that gives you oscillations, as it turns out. Not a surprise. Sorry. I mean, I guess I'm stating the obvious at this point. But this one desperately needs software to help us with it, and the software doesn't exist yet.
I have a quick naive question, if I
Yeah.
can. That's very, very naive. When you say fragments, is this any connection to the fragments that is done in sequencing?
No.
Good.
Okay.
Thank you.
No.
I'm
Yeah.
relieved.
These.
I feel
Yeah.
better.
Yes. The fragments I'm talking about, they are again, they're kind of subsets of the overall bipartite graph of the system.
Okay,
Um,
fun.
so I'm going
Thank
to get into an example now, in fact, because everything so far has been a little abstract. But again, I'm trying to kind of give you a bit of a sense of the sorts of things. So a few years ago, we started working on a model of left-right development. And it's a really interesting system. So this is something we don't often think about unless, I guess, unless you're the type of person who does a lot of surgery on animals. But on the outside, we look relatively symmetric, but on the inside, we're really not. And the interesting thing is that the internal asymmetry is actually fairly reproducible. The vast majority of humans have their heart leaning over to the left and the right lung's a little bit bigger. And, all the internal organs are placed in fairly specific places so that, when a surgeon's trying to figure out if you've got appendicitis, they know what side of the abdomen to poke at, for example, right? Um, and people who have perfectly inverted internal organs, they do exist. It's called the citizen versus, and but they're relatively rare, and most of the people who do have some sometimes subtle and not terribly dangerous, but some defects in often in things like the way the heart is the plumbing into the heart. So the way the blood vessels enter the heart and where they go from there. So um, most of the people who work in this field talk about two particular proteins, lefty and nodal. And nodal is it's part of the TGF beta family. Um, and it binds to a cell surface receptor, which in its internal domain is an enzyme that phosphorylates SMAD2. Um, a couple of SMAD2s and a SMAD4 get together and they make a complex. We're not exactly sure of the order in which they do this. In fact, the fact that it's two SMAD2s, if you ask people in the field, they'll tell you they're not even terribly sure about that. This complex translocates into the nucleus. There's other proteins involved, but and a really complete model would add even more proteins, but we started here because these seem to be the key ones. This thing, plus the other accessory proteins that get involved, what it does is that it's an activator of gene transcription for some specific genes. And in particular, that complex activates the transcription of nodal. So there's a positive feedback loop here where nodal binds to a cell surface receptor, and then it activates transcription of itself. But it activates transcription of another protein called lefty. Now you can guess from the name that when lefty was first discovered, it was discovered on the left side. And in fact, at first people thought lefty was the thing that made that determined the left side. But it turns out that's not what lefty does. So what lefty does is it's an antagonist of nodal. It binds to the same receptor as nodal and stops it from binding. And nodal activates not only its own transcription, but it activates the transcription of lefty as well. So for those of you familiar with these things, that suggests maybe a Turing kind of mechanism, right? You have a self-activator and you have an inhibitor that is that's activated by the same activator. It looks an awful lot like a Turing system. And so that was one of the things we wanted to think about when we got involved in this is, do we have the right biochemistry here? Would it give the kind of behavior that we need to get Turing patterns? And I should say there is a competing hypothesis, which is that it's not a Turing mechanism, it's actually a wave propagation mechanism, and there's kind of a wave of nodal. So it's a fairly, and I don't have every single chemical species in this diagram. So in total, the model has 18 distinct chemical species. there's 5 conservation relations. So this is things like the active gene plus the kind of inactive gene that doesn't have the transcription factor. That there's a fixed amount of that in a cell, right? Essentially 2 copies, right? Um, uh, because of the time scale over which these things happen and what we think is known anyway, we treated the total amount of SMAD4 as a constant. So there's a conservation relation associated with, kind of SMAD4 all by itself and SMAD4 in the complex, that kind of thing. So there's a handful of conservation relations. So when you take those things into account, the number of independent chemical species, so the number of chemical species minus the conservation relations, is 13. So it's a 13-dimensional model, if you will. Okay, so this model, it's pretty easy to find conditions under which it's bistable. And here I'm showing just one of the bifurcation diagrams we looked at. So we looked at things like, would leaky expression of nodal matter? And the answer is, well, yeah, if the leak is big enough. So this is what's supposed to be the inactive form of the gene, and it's making nodal. And if you make the rate constant for that big enough, eventually you don't have bistability anymore. So you have bistability. I should say. So the solid curves are stable steady states. So from here to the left, we have 2 stable steady states. This dotted curve is an unstable steady state. Essentially, it acts as if this was a 2-dimensional model, it would act as kind of what's called the separatrix between getting to the lower steady state versus getting to the upper steady state. But this is a multi-dimensional model. It's a little more complicated geometrically, but in any event, yeah, you get bistability, but if the leak is too large, you don't, which is not a big surprise. Because then you lose control of nodal, right? It's not self-activating. You're just making nodal all the time. Um, you can pick almost any parameter in this model and generate a bifurcation diagram that has one of these nice little saddle node bifurcations somewhere. But the interesting thing, again, is that this particular wiring seems to pervasively give you bistability. So then the question is, okay, well, what parts? Again, it's a big model, right? What did I say? 18 chemical species. Which parts of this model are really essential? Which parts do I need to get this behavior? And the answer is, there it is. So this is the smallest critical fragment we found. And now I'll close the loop to this business of for a saddle node bifurcation, I should have a critical fragment of order, in this case 13. This one's not 13, it is 7, I think. 1, 2, 3, 4, 5, 6, 7. Yeah. So this is a critical fragment of order 7, which, if you think back to the theorems I told you about a couple minutes ago, says this thing should have Andronov-Hopf bifurcations, and if you look hard enough, you can find some. You can get Andronov-Hopf bifurcations by poking around the parameter space and looking for stuff. But let's look at what this thing is. So here's nodal right here. And so we have a reaction here. This reaction 1 is nodal binding to the receptor, and this is now an active receptor that's able to phosphorylate SMAD2. This little piece here, plus this arrow over here, this is the receptor making a receptor SMAD2 complex, giving us a phosphorylated SMAD2. And then the loop back, that's because, of course, once you've released phosphorylated SMAD2, we didn't deal with the reloading of the receptor with phosphate. We assume that's fast. It would be more chemical species if we did that, obviously. But this is basically Michaelis-Menten kinetics. This little corner here, this little corner down here, this is Michaelis-Menten kinetics with that receptor transforming SMAD2 to phosphorylated SMAD2. Then you take 2 of these guys and you make the phosphorylated SMAD2 dimer. I should say, again, we don't know the order in which these things happen. So it's possible that one phosphorylated SMAD2 assembles on a SMAD4, and then the other one joins. So it's possible that SMAD4 acts as a scaffold. As I recall, it's been a couple years since we did this, but as I recall, we tried various possibilities, and they all gave us more or less the same answer, both as to what the critical fragment was, and it didn't really matter what the order of binding was, as you might expect. Okay, so then this dimer of phosphorylated SMAD2, that reacts with a SMAD4 and you get your transcription factor. Again, the exact order here probably doesn't matter. Not just probably, doesn't. And then this transcription factor binds to the gene, gives you an active gene, and then the active gene gives you nodal. So, and if you kind of follow this, I'm following the arrows all the way around here. So there's a big positive cycle there. And then there's some little ones too. This is a positive cycle, and this one's a positive cycle as well. And so this thing has, I think just those 3 positive cycles. I don't think there's any other way to circulate around this thing. Um, so this thing has, the odd number of positive cycles that you need. And as it turns out, it's critical. I can't remember what the value of K is. It's minus. Actually, I do remember. It's -2. It turns out this coefficient here, again, regardless of which order you put it in, that turns out to be an important aspect. So we got 2 pieces of information here. One, what's the instability generating fragment? Right? So this is it. This is the core, right? This is the thing that gives you the interesting behavior. And 2, you really do need, because this was a debate in the literature, you really do need 2 phosphorylated SMAD. If the rest of the framework is more or less okay, you really do need 2 phosphorylated SMAD2s as part of that transcription factor. One of them will not do. If you reduce that coefficient to 1, what happens is instead of a saddle node giving you bistability, you get a transcritical bifurcation. So the fragment is still critical, but the type of bifurcation changes. And that we can do a little bit of math behind to kind of show that. I think I saw something pop up about something in the chat
Yeah,
again.
there's a question. So, and Angeline Lau, you want to
Yeah.
say it or? Okay,
Yeah.
she says, I think oh, there you are. Good.
So, because based from how you illustrated, you were trying to visualize, or you're trying to determine whether the state or the properties, topological properties of whether there's some stability or oscillation exists based on the nature of the graph. But I think what if the network becomes more complex or you have more nodes? Will it be easy to compute or to identify whether there's a cycle, multi-stability?
So the lovely thing about this method is it's very parallelizable. And there's a piece of software that Maya and some of her collaborators wrote that finds critical fragments, and you can distribute it. If you have a multi-core computer, you can distribute it. You can run multiple instances, one for each core. If you have a network, you can distribute it over your network and use a cluster, or they don't even have to be particularly closely coupled, in fact. So it's very, very parallelizable. So yes, you can do larger examples. And we haven't pushed this to see how far you can go. To be honest, a lot of the models we work with are in kind of this size range of 20-ish chemical species. Maybe 15 or so of them independent. But I don't see any reason, if you have reasonable computing capacities, why you couldn't do a model 10 times as large. It would take more time, obviously. Even distributing it over a network, it would take more time, but it would be quite feasible.
For example, in chemical reaction network theory, they try to analyze the topological property of network based on the stoichiometric matrix. And from there, they try to see whether, based on the deficiency theory that Feinberg's actually stabilized. So if it falls under deficiency 0, then you have the existence of a stable or steady state, or a stable stabilization in that given state. And if you have deficiency 1, then there's a possibility that the existence of multi-stability.
Yeah, and there's theorems too that tell you if you have deficiency 1, but then there's some auxiliary conditions, then you can rule out multi-stability. Yeah, they have. It's quite a well-developed area, and it is. To my knowledge, anyway, we haven't run any speed tests. I think it's about as much work to do the, if you want to go beyond kind of the deficiency, the simple deficiency 0 and deficiency 1 theorems, because there are kind of additional tests they apply. It's about as intensive as our method, but I don't want to swear to that because we haven't really run any tests. All right. Okay, now the other thing that was interesting, and so let me just run around this real quick. So here's nodal. Then we've got the signaling system with SMAD2. This is the nodal gene over here. There's no lefty in here, um, and so the you can in fact completely remove lefty from this network, and in that case, if you do that, it turns out that this critical fragment, its order exactly matches the number of independent species in a nodal only model. And so a nodal only model is bistable. And you can draw a bifurcation diagram and what have you, and and see that. So a nodal only model has the capacity to be bistable. Now that was a slight surprise to us because everybody always talks about the nodal lefty thing being important, and lefty um, being a Turing morphogen, it's the inhibitor in the Turing thing. Um, but it turns out there was an experimental result that we found out around the same time we were working on this, that that backs that up. So what they did was they disabled the lefty gene in a cell line. I can't remember what cell line it was, and then they looked to see if they could get the development. So this is it's an artificial in vitro development system, and they looked to see if they could get left right uh, development. So, nodal on one side and not on the other. And it wasn't very robust, but they could, and it got better when they added lefty exogenously. So now, if you think about, so the classic kind of paradigm was lefty and nodal are produced together. They're a Turing pair, and so what you're getting is, again, that kind of classic kind of Turing short range activation, long range inhibition thing. I should tell you too, lefty diffuses faster than nodal through tissues, as it turns out. So that was kind of the operant kind of theory in the thing, in the field, because it looks like a Turing pair, right? But it turns out you can take lefty away, and you can still get left-right development if you add a little bit of lefty, but uniformly over the whole thing. So the control of lefty by nodal is completely non-essential, and they're now thinking maybe what lefty does is kind of titrates the nodal response. So that was interesting too. Um, yeah, so this is k equals 7, which is less than n, but if I take away lefty, then k matches n, so we get bistability there. Okay, so a model without lefty can be bistable or generate, we haven't actually done the Turing stuff, but it, at least in principle, can give you Turing patterns. A model with lefty can have Andronov-Hopf and Turing bifurcations because you have a critical fragment of order less than n. And this is a thing that it makes this theory kind of interesting. And we've now seen this in lots of, well, I mean, the theoretical possibility was always there, but now we're starting to appreciate this a little more. If you have a small critical fragment, you can make it into a bigger critical fragment by adding some stuff that doesn't mess up your critical fragment. So a simple thing to do is just add an edge that's not connected to the rest of the fragment at all. And if you have a big enough model, there's always ways to do that. So in other words, if you have conservation relations in particular, it always seems to be possible to do that. Or maybe you add another little cycle or something. And so your small critical fragments tend to be the root of a family of critical fragments of increasing k. And you can go all the way up to n. And that's what happens in this model is there's a whole bunch of critical fragments of order 13, and many of them trace their origins back to this one, which is just what it says there. All right. So one more trick I'll just mention really briefly. So if you make one small change to the way we calculate that K coefficient of a fragment, you can get a condition for delay-induced instabilities that looks very similar to what you get for differential equations without delays. And so I find that really interesting. It leads to a necessary condition that's a little bit different. So you have to have at least one subgraph within the fragment that has an odd number of cycles of which an odd number are negative. And so you can start to look, I mean, in smaller models, you can look for them by eye. In bigger models, we probably need software. We need to tweak Maya's software to do this. So really briefly, so this critical fragment business, I wouldn't call this software 100% complete, but it's usable. We use it. So there's the software called Graphical. And by the way, I've put all these software into the spreadsheet. So if you guys want to go find it there, it's there. So there's a Python program called Graphical that Maya and her friends developed. If you are interested in the chemical reaction network stuff, there's a couple of resources. There's a web-based one called Control, which runs around and applies pretty much every, I shouldn't say pretty much every, but it applies a heck of a lot of tests that come from this chemical reaction network theory, and just kind of spits them out on your screen after, it runs around, it sends stuff off to its server and then spits the stuff out. There's an older piece of software, and to be honest, I've never kind of looked super hard at, I've used both at various times just playing with them. I've never looked super hard at kind of whether they do different things, but there is a Windows-based program that you can get from Martin Feinberg's website called the Chemical Reaction Network Toolbox. And I don't know how much of, I don't know to what extent, I know they overlap, but I don't know whether one of them does stuff that the other one doesn't. I've never kind of looked super hard at it. And just to finish this off, so I've mentioned Maya a whole bunch of times. She's a faculty member at Northern Illinois University and has been for some years, doing some really interesting work over there. Catherine Roussel is both my wife and my collaborator. We work at the University of Lethbridge in Alberta, and NSERC pays the bills. Yeah, so that's it.
Thank you, Mark. I definitely learned a lot. Does anybody have specific questions? I actually, just because this is definitely out of my field, and I just wanted to, I definitely didn't know what to expect, but I still was curious about, so I'm guessing the core was this final fragment that you cannot shrink any further.
Well, okay, so
I hope I understood it.
you can't take away anything from that fragment, yeah. So it's the smallest piece of the model that generates instabilities as the model is now. But one of the things we're thinking about, so the idea of shrinking it is interesting. One of the things we're starting to think about is, well, can you use this theory then to come up with effective ways of reducing a model? Essentially, so a lot of these things, you often end up with a great big cycle, and you don't need all of that to make the fragment critical. If you can cut some of the, kind of, you can think about, a cycle with maybe, six species in it, and take a couple away and glue it back together, and it still works. That corresponds to model reduction where you squeeze out some intermediates effectively. And so it may be possible to use these methods to do model simplification in addition to just finding these critical pieces, these critical fragments.
Thank you.
So I just have one question. That's it. So thank it was a really informative talk, and I really enjoyed it. So I was wondering, you mentioned the order is not really important. So why is that? When you showed the smallest critical pathway, so you mentioned the order is not really important. So
it is and it isn't, right? So the fact that I have a critical fragment whose order is less than n means that I can get an Andronov Hopf bifurcation and oscillations. But once I've found that, I can make bigger fragments that are still critical by adding edges and small cycles to it. And so again, that little critical fragment kind of blossoms out into a whole family of critical fragments as you increase the size of the fragment. There are other critical fragments. So in this particular model, there's a heck of a lot of critical fragments. There are other critical fragments that kind of originate at larger Ks that also kind of blossom out into a family that keeps going all the way up to n. So it's not so much that it's not important as it turns out again that if you find a small critical fragment, I can almost guarantee there's going to be critical fragments all the way up to the end of the sequence. Because again, it's pretty easy to take a critical fragment and add something that doesn't mess up the math that makes it critical.
So how can we understand that if we add something and it doesn't make it critical?
It doesn't change the fact that it's critical. Yeah. So it has to do with the structure of the so it has to do. So if you think about when I'm going to develop the characteristic polynomial, I need to take the determinant of kind of lambda I minus j, right? And how do you take determinants? You compute these minors, right? So you delete rows and columns, and then right. So you've got so that process ends up in many of the steps of that process, what you end up doing is taking a piece that you already had at a lower order and then multiplying it by, essentially one of the J's from the Jacobian. So if that piece before gave you something negative, just multiplying it by something, as long as it's something positive, which most of the bits in the Jacobian often are, then that doesn't really change the fact that that piece was negative. So the kind of the negative terms tend to propagate up through this calculation. I don't know if that makes sense.
Yes, kind of. Thank you.
It's and all of this stuff, like if you look at how this stuff works, I mean, ultimately a lot of this stuff rests on, in one way or another, on Ruth Hurwitz, right? And in Ruth Hurwitz too, if you kind of apply that to models that aren't too big, so you can still do the calculations, you tend to find that when you find these I think they're negative minors in the Ruth Hurwitz method, that once you find one, you get the higher order Ruth Hurwitz minors tend to also kind of retain that sign, for the same reason that if at some point you're basically just multiplying something that's already negative by another little positive piece.
Yep. Thank you.
Yeah.
I have a question, if I may.
Sure.
The lefty nodal model was pretty big, right? And you were saying when you're showing the bifurcations that you can pretty much pick any parameters and you'll get a bifurcation, if I remember correctly.
Yeah, once you identify an area where there's biastability, then fooling around with the parameters, sooner or later when you fool around with the parameters, you'll go through a bifurcation and lose the biastability. But the nifty thing about this model, I mean, the thing that gives me some confidence that probably this model has a lot of details right, is that the biastability exists over a really wide range of parameters. Like you can bend the parameters all out of shape and still find biastability, which is good because we don't know what the parameters are. Anyway, I think
I was going to ask about that. How did you decide what the parameters were? Like, did you find literature where
no,
has some inclination to?
So exactly, that's exactly the problem, right? There's nobody who's measured any of the rate. We cannot find a single rate constant for this mechanism. We can't find association rate constants for phosphorylated SMAD2s, catalytic rate constants for, that receptor phosphorylating SMAD2, nothing. So all of the work that's out there is looking at the connections, the actual wiring. What are the reactions? What associates with what? What catalyzes what reaction? And that's pretty much, and then of course lots of developmental experiments, right, where people look at, if you mutate this or cut that or whatever, what does it do? So when we went looking for parameters, initially, well, again, because it is a model that does seem to want to be bistable, it wasn't too hard. We essentially kind of, we did the old-fashioned thing at first, where you just kind of eyeball it and think, well, I probably need, this particular. I mean, we'd already kind of spotted there was a positive reinforcement loop, because that's kind of the whole point of the model. And we just kept saying, well, maybe you just need to make that a little stronger and tweak a couple of these rate constants. It took us very little time to find rate constants where we had bistability.
I guess my follow-up question is, how do you actually play with the parameters then? Do you just kind of pick one and go with it? Because if you're trying to mess with like 10 different parameters, that's a lot of different possibilities to work with. And I mean, I guess you can try plotting them, but you only have 3 dimensions, right?
Yeah, most of the people I know who do this kind of, so there's maybe I'll answer that in 2 parts. So the first one is just the practical one. So most of the people I know who do this kind of work, whether it's developmental or in other areas of cell biology, we tend to do very stupid things at first, which is we set all the parameters equal to 1, and then we just see what it does, and then inevitably it doesn't do anything interesting at that point. And so what you do is, again, you kind of ask yourself, well, what do I think is going to be the stuff that's going to be important here? So do I think that inhibition by lefty is going to be important? I probably did think that at the time, right, because we didn't know kind of where the critical fragment was. So we probably pumped up some of those rate constants to make that more effective. And then, we probably found, oh, gee, now you don't get a whole lot of nodals, so pump up some of those rate constants. And, again, I'm not sure we even spent as much as a day looking for parameters. Like it, and this is the second part of my question, of my answer, is if you talk to people, so you talk to John Tyson and his cell cycle modeling, and, just any of a number of people who do these kinds of things. If the wiring is right, generally speaking, the behavior is really easy to find. If the wiring is wrong, you'll never get anything out of it. And so when you, and that makes sense. If you think about the conditions under which biological systems evolve, if the behavior was excruciatingly dependent on the parameters, nothing would ever work, because even a minor mutation in a promoter that changed the rate of expression of a gene, or a mutation in a protein that slightly changed its binding constant to something else, any of those things could mess it up. So in fact, they have to be wired in such a way that the region of parameter space where you get the behavior that gives you whatever behavior it is that's important to that system has to be really large. So that would be my answer to that. And the reason why, again, it's not that hard to find parameters that do qualitatively the right thing.
Yeah, heaven forbid you get a gene duplication event, I guess.
Well, and yeah, and that's, without going to a gene duplication event, what if you inherit a mutant allele from a parent, so only one of your gene copies is any good? So you a lot of critical, not all of them, right? I mean, certainly there are haploinsufficiency diseases, right? But an awful lot of things are actually robust to losing half of the gene expression. And sometimes they're robust just because, again, your normal level of expression is right in the middle of a great big region of parameter space. So even if you move quite a ways inside that region, you're still okay. Sometimes they're robust because the control mechanism, the feedbacks compensate for losing a working copy of a gene. So you just, that one gene that you've still got is just on more of the time to compensate for the one that's never producing any protein.
I guess I'll let someone, thanks for that. I guess I'll let someone else ask a question, but I have one more.
Yeah, I have another question, and this is again naive. Can you deal with the parameter problem with machine learning?
Yeah, I would think so. We've never done that, but I mean, there's a lot of these things where I'm sure you could use machine learning to automate some of the stuff we do, and certainly, kind of picking parameters that give you a desired behavior. I don't think that would be, relative to the things I've seen people do with machine learning, I don't think that would be even all that difficult. The danger is that the machine learning might be too good at it, right?
Yeah.
So one of the things that we find when our models are not quite right, um, close but not quite right, is that you get a really small region of parameter space where you get the behavior you're looking for, right? So if you're missing some key, feedback or something like that, you might still get the behavior, but it's in a small region of parameter space, and we tend to have trouble finding those. Once we've identified critical fragments, of course, it's pretty easy then to fudge it, because we know what term of the Jacobian we need to pump up and what it looks like, right? But when we're kind of poking around, we'll often miss these kinds of things, and that's sometimes a sign that there was something we should have put in the model that makes it more robust. Whereas a machine learning algorithm might just be really good at zeroing in on these tiny little regions of parameter space and giving you parameters where the thing will do what you want it to do.
Okay, great. Now, usually there is a problem with the huge number of parameters, right?
Yep. Yep.
Now, it may be too wild to think like that, but maybe dimension reduction of the space of parameters can be useful here.
Yeah, and there's people who work on those sorts of things. And I think some of them do use, now that you mention it, I think some of them do use machine learning algorithms to kind of find a manifold in parameter space where certain things happen.
Right.
Yeah. Yep. Alexander Gorban in Leicester, I think, does that kind of thing, for example.
Where is he?
Leicester in the UK.
Leicester. Okay.
Yeah. Yeah.
Think I came across the name at least, if not the application. Anyway, thanks.
Yeah. Well, he does a whole bunch of different things. So you could have
Right.
run into his name in any of a number of contexts. Alexander is one of these
Yeah.
kind of wonderful, crazy people who's totally not afraid to do whatever runs into his area. He's done some stuff on, what was he doing last time I talked to him? He was doing some stuff on computer security, like using AI to figure out if your system's been hacked. So, he does all kinds of crazy stuff. Great guy.
That's good. It's good to be wild type.
Yeah, exactly.
Okay, thanks.
I think Serge had another question. I don't know if somebody else does.
Yeah, since no one's spoken up, I guess I'll ask mine. This is a pretty naive question because I'm not very, I'm familiar with critical points and things like that, kind of, and the network theory is pretty interesting. But when you're showing the network and you're talking about reducing things, I was kind of drawing a parallel to typical algebra, right? Where you have x can equal y plus z. I was wondering if you could reduce, for example, one of these little positive cycles to some other representation to simplify your network, basically, so it's easier to compute.
So that's one of the things we're trying to think about in terms of using these graphs to figure out what interactions we can simplify. So for example, the little loop that I said, hey, this is Micheli's-Menten in here. That's what it looks like in these things. You retain, the fragment continues to be critical if you replace that by what you might call direct or contact catalysis. So in other words, instead of having enzyme plus substrate makes a complex, makes enzyme plus product, if you just go enzyme plus substrate makes enzyme plus product, that does not affect the criticality of the fragment. Now, what it does do, though, is it messes with the region of parameter space where, or it may anyway, mess with the region of parameter space where stuff happens. And that one is particularly pernicious because you lose saturation, right? So if you have an enzyme substrate complex, then there's a maximum rate at which that reaction can be catalyzed. Whereas if you've got E plus S goes to E plus P, it's just linear in S, no matter how much S you've got, right? And so that can change the size and shape of the region and parameter space where interesting behavior happens. We don't have a good handle on that yet. We are thinking about it, but that's a kind of a future direction for us, because kind of going from saying, okay, that's a critical fragment, to saying, and if I change this, here's the region, here's what happens to the parameter region where you get bifurcations. That's not a, that requires another layer of theory we haven't quite developed yet. I have some ideas as to how we might do that, but we're not there.
I think I got some of that.
Okay.
I was thinking also
I
that
guess, sorry, maybe
on.
one more thing, which is, of course, the other thing you can do, and people do routinely, is, you use the steady state approximation or something, right, to simplify some of these things. But then you're going outside of mass action, and then you need a slightly different set of theoretical constructs to do these analyses. So you can't use exactly this piece of theory. And from the little bit I've done with kind of the non-mass action stuff, it seems like the theory is not quite as powerful when you go outside of mass action. I say that, and probably George Crasian or somebody like that will have developed something brilliant, in the next few months that will totally blow this away. But at least so far, anyway, what I've seen, it's not quite as powerful.
I think that's it
Thank
for
you,
my
guys.
questions. So.
I'm gonna actually have to go. We're quite over time. Sorry. I made you the host.
I tried to make not too long a presentation, and somehow it got away from me.
It was an hour. You broke my rule.
I did. Okay. I'm bad.
It's okay. I didn't want to interrupt because it looked like nobody was asking questions, but it's just over time. You guys are welcome to keep on talking, but I do have to run to another, or switch to another meeting.
Yes.
And hopefully I'll see you guys in 2 weeks' time. Do you guys still want to talk about, I guess I should have asked everybody else, but I think we said that if we can simulate a perfect cell, will it still be useful, or do we want to do something else? Like today we were talking about figuring out parameters. That's also another topic. Yeah. So are you guys okay with the next topic? Yeah.
I like
Anybody want to lead it? No, nobody ever wants to lead it.
Well, I think that's a good one to talk about. I don't think it needs a complicated,
okay,
kind of,
good.
Right?
So I'll set it up. It's just always hard to. Okay.
I'll try.
I would be sorry.
Go ahead.
Maybe everyone could just submit their slides on the topic, and then Maria, you just collect them. Sorry to burden you with that, but
no problem. I played that game before. It never goes well, trying to speak on somebody else's slides. But yeah, I mean, you guys are welcome to give me whatever information, and I can try to make something from it. But also, I guess I'll just do a quick introduction next time and hope that people can just roll from there. Is everybody okay with that? Great. And I thought I heard another voice.
Yeah, I was saying I would be interested in talking more about the parameter estimation, like maybe just on that topic, still new to all
yes,
this.
I think it's on the list of potential discussions, and I think Zoe was one of them who was willing to, and I guess Mark, you could be also one of the people who can show how somebody would find parameters. So I think that's definitely an interest. And please add more discussion topics. Things also show up when we're discussing here anyway, so we're always
very informative talk. Thank you.
You're welcome.
To contribute estimating parameters as well. Me, like specifically these days, the models that I use both like as Mark said, like just let's give one to everything. And also I use some parameters from the experiments. So I would be happy to also contribute on that topic.
That would be great. So if you guys, we can coordinate. I mean, the next meeting is available is May 25th. So if you guys are okay with that, I can slot that in. I just need everybody to, like, if you're willing to do it that day, just confirm or maybe put your name and then I'll know that that's good to go, if that's okay with everybody. That would be great. And thank you guys very much. I really enjoyed this and learned a lot. Okay, bye.
Bye.
Right?
Thank you, Mark. It was
Yeah,
great.
that was an
welcome.
interesting talk.
Yeah, thank you.
Bye. Thanks, everybody.
See you next time.